Food security is an urgent global issue affecting nearly 10% of the world's population, with over 400 million people in Asia suffering from hunger. Soybean, as one of the most valuable cash crops in the world, has the potential to address this problem by providing a major source of protein and oil for human consumption and animal feed. In fact, soybean is an ancient crop that has been cultivated for thousands of years, and the earliest known record of soybean cultivation dates back to the Book of Songs, a collection of Chinese poetry that is over 3,000 years old. However, over thousands of years of domestication, cultivated soybean diversity has drastically decreased, leading to the loss of some important traits, for example, stress-tolerant traits. Therefore, mining the genome of wild soybean for gene discovery is a new strategy for crop improvement. Aspiring to bring soybeans back to China, Professor Lam from the School of Life Sciences identified and cloned a novel salt tolerance gene from wild soybeans with genome sequencing and molecular biology technology, and applied it to the breeding of salt-tolerant soybeans using molecular markers. But how does that actually work? First, the research team selected two soybeans, W05 and CO8, as parents for breeding. W05 is a wild soybean with high salt tolerance, and CO8 is a cultivated soybean sensitive to salt. By crossing these two soybeans, they created a recombinant inbred RI population. Followed by self-pollination of the F1 generation and F2 generation, single seed descents from the F2 generation were then obtained and planted, creating a nearly homologous RI line population. Out of around 500 RI lines, 96 core lines were selected to undergo low-depth whole genome sequencing (WGS), an efficient and cost-effective technique that allows population-scale screening of the entire genome while retaining individual information, including single nucleotide polymorphism. A bin map was then generated from WGS. A bin map is a high-resolution genetic map that divides the genome into small non-overlapping intervals, or bins. Each bin is defined by a recombination breakpoint and contains a collection of SNPs or other polymorphisms. By using the bin map and phenotypic data such as their growth period, stress tolerance, and coloration, etc. from the RI lines and the parents, genomic regions associated with the trait of interest can be identified. With this method, Professor Lam identified 11 agronomic traits with major quantitative trait loci, QTLs, which is a region of DNA that correlates with a particular phenotypic trait that can be attributed to multiple genes as well as environmental factors. The research team then focused their subsequent analysis on the salt tolerance locus on chromosome 3 in W05 and discovered that the GM-CHX1 gene is associated with salt tolerance. They revealed that GM-CHX1 is important for maintaining a lower sodium-potassium ratio under salt stress, and that the loss of function in CO8 contributes to its salt sensitivity, while the normal function of the gene enables W05 to be salt tolerant. Professor Lam wishes to contribute to environmental sustainability through transferring his research findings to practical use on farmland. In partnership with Professor Zhang Guohong of the Gansu Academy of Agricultural Sciences, he developed salt and drought-tolerant soybeans by integrating molecular markers and traditional breeding. Among the soybeans developed, Longhuang No. 1, 2, and 3 have been approved for field application in Gansu Province of China. Since 2016, the cumulated acreages of these varieties in semi-arid areas in the Loess Plateau have exceeded 830,000 mu, showing a significant improvement in the yield and quality of soybeans. Apart from collaborating with mainland China, Professor Lam wishes to benefit smallholder farmers in other developing countries. Recently, Professor Lam has initiated soybean cultivation programs in South Africa and Pakistan in hope to help them increase their soybean harvest yields.